Hello friends, welcome to EduTap and also welcome to the series which we have started for IBPS AFO 2020-21. That is for that too for mains exam, that is agriculture, and this is lecture number 43. And as per your suggestions, I have taken up today one important topic, that is genetics and plant breeding, in which we are going to do few MCQs, not 5 this time. From this we are going to do 10 MCQs, and first we'll be touching few basic genetics, and then slowly we'll move on to plant breeding and what are the various concepts related to it in the form of MCQs only. So friends, before getting into course details and various other course offerings, let me take you through our success stories which we have achieved since the past few years. And these are the students who have achieved their dream of becoming AFO during last year, that is IBPS AFO 2019 exam. And not only that, we have students in top 10 all India ranks as well, and we have few students in top 20 as well, and we have many students who are in top 50 all India ranks and achieved their dream of becoming an AFO. Friends, I have an important announcement here. One, as you all know, we have launched courses for IBPS AFO 2020-21, and the links to the demo courses as well as to the payment towards the purchase of the courses has been already mentioned in the description box along with this video. And along with that, I would like to bring to your notice that we have launched a discount coupon for these courses, and the coupon code is AFO50, and we are offering a whopping 50% discount on AFO courses. So if you or any of your friends want to purchase this course, this is the right time. You can go ahead with that using this code AFO50, wherein you'll be getting 50% discount. So now let's start with the session. So friends, here comes the first question. Which of the following pairs is or are correct? Nucleus, controlling center of cell; mitochondria, powerhouse of cell; ribosomes, site of protein synthesis; lysosomes, suicidal bags of cell. All these are correct. And one more organelle I'll tell you, that is chloroplast. This is also called as kitchen of the cell, because you all know this is where photosynthesis occurs, and that is where preparing the food. That's the reason this is also called as kitchen of cell. So let's see the options, whatever they are mentioned there. Nucleus, yes, it contains chromosomes and genes which controls functions of cell, and this contains mainly three parts: nuclear envelope, nucleolus, and chromatin, the genetic material. Mitochondria, yes, this is called the powerhouse of the cell, and it's a rod shaped cytoplasmic organelles, and it's a main site for cellular respiration, and they are the sources of energy. This consists of three main parts: membrane, cristae, and matrix. And the third, ribosomes, these are the smaller cellular particles, as you can see here and there. And then these are the main sites for protein synthesis, and these ribosomes, they are rich in RNA, ribonucleic acid contents. Lysosomes, these are also cellular particles that contain several digestive enzymes, and the main function of lysosomes is digestion of intercellular substances and any foreign particles that attack. And then when the cell dies, it releases its enzymes which digest the dead cells. Hence, that is the reason they are known as suicidal bags of cells. So friends, crossing over takes place in which stage of meiosis? See, in the meiosis it occurs in two phases, and in phase 1 in meiosis this stage occurs wherein crossing over occurs. What is that stage? That is Packetine stage. So friends, Packetine is the stage where crossing over exactly takes place, and if you need to understand that entire one, I just give you a brief of it. Meiosis as such occurs in two phases: meiosis 1 and meiosis 2. And meiosis 1 has four phases. What are they? Prophase. Since it is occurring in meiosis 1 first phase, it's called prophase 1. Next comes is metaphase 1. Next comes is anaphase 1 and last is telophase 1. Now in each of these phases various functions takes place and various specific functions takes place, thereby the entire meiosis occurs wherein the diploid cell, it gives rise to, one diploid cell will give rise to 4 monoploid cells. 4 monoploid cells are produced. Here meiosis, here DNA replicates once but nucleus divides once, thereby giving rise to 4 monoploid cells, unlike in mitosis where only 2 cells are produced. That too from diploid, again same diploid will be produced, but here it's different because these are generally, this kind of meiosis division takes place in reproductive cells. Okay, so now this prophase, it is further divided into various other phases. Leptotene, they are leptotene, zygotene, pachytene, the question on this is asked, diplotene and diakinesis. Now pachytene is the stage where crossing over or exchange of chromosomal or genetic material takes place between non-sister chromatids, and this, as we have seen, the stage of prophase 1 in meiosis, and formation of chiasma and crossing over takes place during pachytene. Chiasma is nothing but it's a place or it's a point where non-sister chromatids cross over. Chiasmata is generally plural and chiasma is singular. And in this phase, generally the chromosome look bivalent in nature. It is have 2 strands. So they look similar to that bivalency nature. So this is about the pachytene stage which occurs in meiosis, in again meiosis 1. So friends, here comes the next question. The physical or chemical agents which greatly enhance the frequency of mutation. Mutation is anything sudden change in the genetic structure or chromosomal structure. It is called mutation, and this is called as the physical or chemical agents that increase, that is called as mutagens. So friends, mutations can occur based on, because of various reasons. They might be, physical mutagens or chemical mutagens, and sometimes even biological mutagens also, like viruses, bacteria, etc. They can also cause mutations. And here the physical mutagens, they include ionizing radiation, where ions, they are involved here, like for alpha rays, beta rays, gamma rays, X-rays, etc. And non-ionizing radiation, other than ions that are because of which their mutations are created, that includes especially UV rays, ultraviolet rays. And then chemical mutagens, there are many types of chemicals that can create mutations, and they include alkylating agents, which include the mustard gas, EMS, that is ethane methyl sulfonate, etc. Base analogs, 5-bromouracil, 5-chlorouracil, acridine dyes, some dyes also, some kind of dyes also can induce mutations, that is acryflavin, profalvin, deamination agents which include ethidium bromide. So these are also various chemical mutagens that can induce mutations in various organisms and plants. So friends, here comes the next question. Which of the following is or are the methods of breeding for self-pollinated crop? One straight question, and here the correct answer is pure line selection. Pure line is nothing but it's a progeny of a self-pollinated homozygous plant, and development of any new variety, through identification and isolation. They'll identify and isolate any single best plant progeny from the existing lines. It's called as pure line selection, and this is generally followed in self-pollinated crops. And here this concept of pure line, it was given by Johansson, which was done by in Fasciolus vulgaris. He was doing some experiments in Fasciolus vulgaris variety, especially Princess. And while working on that, he came up with the pure line concept. And as I mentioned earlier, it is a homogeneous progeny of a self-pollinated homozygous plant. And development of any new variety through identification and isolation of a single best plant progeny, it's called as a selection. Such kind of selection is called as pure line selection, and it is commonly used in self-pollinated crops like wheat, barley, and all the plants that in the pure line have the same genotype, because they are self-pollinated crops. And other than this pure line method, there are various other breeding methods used in self-pollinated crops. They are pedigree method, single seed descent method, back cross method, and bulk method. So friends, here comes the next question. The superiority of F1 hybrids. F1 hybrids is nothing but the filial 1 hybrid. That is, whenever cross is done, X, Y, then whatever the first progeny that comes, this filial, this is called F, stands for filial, and since it's a first generation, it's called F1 hybrid. So the superiority of F1 hybrids in one or more characters over its parents, so it is superior to its parents, it is known as, such kind of superiority is known as heterosis. And the term heterosis was first used by Shull in 1914, and this generally is confined, heterosis, it is confined to the F1 generation of a cross whenever a crossing is done. And the estimation of heterosis can be done in three major ways. One is average heterosis, which means here heterosis is estimated over the mid parent, that is mean value of the two parents is taken, and then heterosis is determined, the superiority is determined. Then heterobeltiosis. Now here, when heterosis is estimated over the better parent, so of both the parents, whichever is better, based on that the superiority of the progeny, that is F1 generation, is measured, that is heterosis is measured. Useful heterosis. Now this refers to the superiority of F1 generation over the standard commercial check variety, whatever is a standard commercial variety is there. So the best qualities whatever are there, on that the superiority of the current crossed F1, the F1 generation would be measured. That kind of heterosis is called useful heterosis. So these are the three kinds of heterosis based on which heterosis can be estimated. So friends, here comes the next question. Which of the following is the most common method of detecting the linkage? Now how it is detected? It is generally done by test cross. Now what is this test cross? See, crossing of F1 individual, F1 we all know now, F1 individual which is having the dominant phenotype. It has a dominant phenotype with its homozygous recessive parent. That kind of crossing is called as test cross, and the progeny of test cross is called as test cross progeny. So X, Y, if something Z is happening, now this progeny is crossed with the homozygous recessive parent, then that kind of cross, it's called as test cross, and the progeny is called as test cross progeny. And now see, test cross is a cross between an organism with unknown genotype and a recessive parent. Linkage is nothing but it's a phenomenon of inheritance of linked genes in the same linkage group. Now this principle of linkage was discovered by Bateson and Punnett in 1906 in sweet pea, that is Lathyrus odoratus. Now however, linkage as a concept was put forth by Thomas Hunt Morgan in 1910 based on his experiment on Drosophila melanogaster. This is one of the important experiment that we have been studying since even in 11th 12th classes also we have studied this, the experiments on Drosophila melanogaster. And There are two major phases of linkage. One is coupling phase and the other is repulsion phase. Coupling phase, it is a linkage between two or more either dominant or recessive alleles. Now these alleles is one of a pair of genes that appear at a particular location on a particular chromosome, and then these alleles are also called as allelomorphs. And here, as you can see, dominant allele, recessive allele, linkage between two or more either dominant or recessive allele. Repulsion, linkage of dominant allele with that of recessive allele, then it is called as repulsion phase. So this is about linkages and how the test cross is done. So friends, here comes the next question. Which of the following is the correct F2 ratio in inhibitory gene action? Now this is about epistasis. Now what is epistasis? Epistasis is nothing but, see, when there is interaction that occurs between different pairs of alleles which influence a character of an individual, then that type of interaction is said to be interallelic interaction. It is called as interallelic interaction, or the same thing is called as epistasis. Now since it has masking effect, now the gene that has masking effect is called as epistatic gene, and the gene whose effect is masked is known as hypostatic gene. Now generally epistasis, it occurs in F2 generation, and the term epistasis, actually it was coined by Bateson in 1909, and various type of epistatic gene reaction interaction occur, of which this inhibitory gene action is one, and the correct ratio of this is that they get is 13:3. So friends, we have seen epistasis, and also we have seen the how the gene interaction happens. See, when expression of one gene depends on the presence or absence of another gene in an individual, it's called as gene interaction. And there are various types of gene interactions. One is complementary gene interaction, inhibitory gene interaction, polymeric gene interaction, duplicate, recessive epistasis, dominant epistasis. So here we got the question, inhibitory gene action, which says that F2 generation they will get 13:3. This is what the ratio it comes out. Now in this inhibitory gene action, a dominant allele at one locus, it can mask the expression of both dominant as well as recessive alleles at the second locus. That kind of interaction is called as inhibitory gene interaction. See, best example here is rice. See here, the gene interaction, it was found that for anthocyanin pigmentation in rice, in green plants, the color in the green color of the plants is governed by gene I, for example, and for purple, that is anthocyanin pigmentation P. So in dominant varieties of green, it would always be capital I, this I, because this is recessive, and here since this is dominant, this can be written like this. So when this is crossed, then in F1 generation, all the plants that they got, they were green in color. So here the dominant allele, it is masking the expression of both dominant as well as the recessive alleles. How they will know that there is some recessive alleles? When these F1 were intimated, then when they cross between themselves, then what they got in F2 generation is 13 green plants and 3 purple plants. So this kind of interaction is called as inhibitory gene action or inhibitory gene action, inhibitory epistasis. So friends, here comes the next question. The progeny of a single plant obtained by asexual reproduction is known as, one direct question, but scientifically it's asked, it's a definition of a. So friends, plants are cloned to produce plants quickly and economically, and the best example of cloning is micropropagation or the tissue culture. Here see, rather than going for allowing the plants to reproduce, then they fertilize seeds and then sowing them and growing them, it takes lot of time. For some individuals, plants which are rare and endangered, for such plants this cloning or micropropagation is the best way of preserving the species. And there are various types of cloning: gene cloning, reproductive cloning, therapeutic cloning. So friends, here comes the next question. Which of the following mechanisms are helpful to promote cross pollination? See, in few plants, in order to promote cross pollination, they adapt to various mechanisms, and such mechanisms they are called as outbreeding devices. And this I have taken an entire lecture on this single topic, because it involves various concepts which I have taken up. And here, decline, dichogamy, hercogamy, heterostyle, these are all the various mechanisms that are adopted by the plant so as to promote cross pollination. See friends, these are the various mechanisms that promote cross pollination in various plants, which include decline or unisexuality, dichogamy, hercogamy, heterostyle. And see, I have already taken a complete lecture on this. I will be taking you through just very fast. So here, decline or unisexuality, it is a condition where flowers are either staminate or pistillate. Again, this decline can be of two types. One is monoecious, where staminate or pistillate flowers, they can be found on the same plant in the same inflorescence. For example, mango, banana, coconut, or they can be found on the same plant but separate inflorescence, like in maize. Dioecious, staminate and pistillate flowers are found in different plants, then they can be called as male plant, female plant, etc. This is papaya and dates. Example, dichogamy. Again, this is a condition where the maturity times of stamens and pistils of hermaphrodite flowers will be different, so that self-pollination can be completely avoided. Now this dichogamy can be of two types. Protandry. Here the stamens mature earlier than pistils. Maize and onion example, protogamy. Pistils mature earlier than stamens. Example, bajra. Hercogamy. See here, it is a condition in which hindrance of self-pollination can be observed with some physical barriers, such as around the anther. Example, in lucerne flower, to avoid self-pollination, they have many layers of stigma. This way they'll avoid self-pollination, thereby they undergo cross-pollination. Heterostyle, different lengths. So here it's a condition where the styles and filaments of the flowers are of different length. This again, because of the different length, they'll not be able to do self-pollination. Again, this promotes cross-pollination, and the best example is linseed. So friends, here comes the last question. Which of the following male sterility is controlled by cytoplasmic as well as nuclear genes? And the type of male sterility controlled by cytoplasmic as well as nuclear genes is cytoplasmic genetic male. So friends, cytoplasmic genetic male sterility is something where male sterile plants, they produce no functional pollen, but yes, they do produce viable eggs. So what happens is, this kind of the Cgms, it can be used in agriculture so as to produce hybrid seeds. Now what they do is, hybrid seed is produced from cross between two genetically different lines. And here such seeds which are produced like this, they result in larger, more vigorous plants. And this Cgms is widely used for hybrid seed production, as we have seen, in both seed propagated species and vegetatively propagated species, because once they cross that male sterile with the help of that eggs with the fertile plant, then the whatever the progeny that is obtained, that generally result in larger and more vigorous plants. And then the Cgms was first discovered by Jones and Davis in onion, and that too in 1944 this was done. So friends, here comes the answer to homework question. In the last lecture, I had asked you a question. What is the pattern of assistance to set up high-tech nursery, which is of 2 to 4 hectares unit under National Horticulture Mission? And here answer for this is the assistance provided is of 25 lakhs per hectare and or 40% of the cost as credit link back-ended subsidy. And now here this is provided under NHM, that is not National Horticulture Mission, or sub schemes of NHM and Horticulture Mission for Northeast and Himalayan states under and also under MIDH, which is nothing but Mission for Integrated Development of Horticulture. So under these three, 25 lakhs per hectare can be provided for setting up of high-tech nursery, which is of 2 to 4 hectares unit. Now today's homework question: what is the cross that is made between two different species but of the same genus is known as? Now since we are doing plant breeding and genetics, question is based on that very easy straight question.